if you missed the very beginning, they talked about the different colors, they may represent different species. These are dead sponges that are being grown on top of. So kind of like coral reefs continue to grow on top of older dead material, these two grow in that same structure. And reefs are important because they provide a habitat for many different types of organisms. All right, so obviously what we're going to cover today, the sponges. We should also be able to hit placozoans at the end of today's lecture. Talk about different aspects of sponges. Before I do, and this is going to be every time we talk about a new phylum in the class, or a new group of phyla, as some of the chapters show, I'm going to ask for you all to contribute in terms of interesting features. Oh, question before?
Yeah. It's just a clarification. Is there anything we need for lab since the first one's on Friday?
Oh, good question.
I know we need a notebook, but is there anything else we need?
Not that I think so. Exactly. That's a great question. Notebook is needed for the first lab this week on sponges. And, you know, that you could have a formal lab notebook with no lines on it to where you could have a spiral bound, you know, set of paper. You could have a stack of paper that you keep in a folder, whatever's going to be easiest for you. And me, I like to have a notebook for labs like this. I know some laboratories require that you have an official lab notebook with these blank pages, no lines on them, so that you can make drawings. But whatever you have for recording notes, not electronic, these are going to be things that you turn in, so something physical pad of paper, sheets of paper stapled together, what have you. But we need something physical to write down observations and responses to some of the directions in the lab handout. Lab handouts, I don't know, they're not yet posted, but they will be posted by this afternoon. So you'll be able to get those and download them or access them on your computer during the lab, whatever is most convenient for you. Good question, though. Thank you. I meant to cover the lab business or course business before I got into the lecture, but completely forgot because I was so mesmerized by the last lecture. Anyways, so based on the chapter reading, something interesting that you learned about sponges, or I think what, they had a paragraph or two on placozoans, or if you've had any direct interactions or experiences with sponges or placozoans, and you'd like to share with class, by all means. Yeah.
Being mobile.
Being mobile?
Yeah.
That they can move to a limited extent?
Yeah.
And what way were you thinking about it?
I did not know that they were mobile.
Okay.
Previously with sponges.
And they can be mobile in the sense that there can be little contractile cells within them. They can be mobile in the sense that, if the cells, they can be fragmented and then there could be movement. The larvae are mobile. Huh? Any other? What else is a cool feature about sponges? Well, we have to have a couple, but I don't want to get many. There's lots of cool facts in chapter reading about sponges, or something, I was trying to think about some personal sponges. I can't exactly come up with it, but sometimes we will for future groups. But anything else from the chapter reading? Nothing? Sponges are so boring? Yeah.
So it's not like Cambrian or possibly Precambrian?
They're from the Cambrian, or possibly. The book kind of put some doubt into that. Possible records going into the pre-Cambrian. So they're a very old group. One more? I know we had at least one more. How do sponges eat? Isn't there something interesting? How do we, when we think about sponges eating, we think about filter feeding, or we should, after reading most of the chapter. But what's another cool feeding ecology that evolved in sponges? And I alluded to this, I think the last lecture when I was talking about choanoflagellates, which are these cells, unicellular, occasionally multicellular, eukaryotes that have these collars and a flagella that allows for them to feed. That sponges have, and I said, or at least most sponges have. Which sponges don't have these choanocytes, these feeding cells that are filter feeding? Oh, yeah.
Excuse my pronunciation, but the Placozoa.
Placozoa, well, placozoans are another group, and they don't have that. So the placozoans don't have, but a type of sponge. So placozoans are completely separate phylum,
Right.
with only well, 3 species now. But within the poriferans, the sponges, that phylum, there's a group that doesn't have any choanocytes, these collar cells that are used in typical sponge feeding.
Is it the demosponges?
Demosponges, they have, they do, I'm pretty sure one of the groups of demosponges is the one I'm thinking of.
Okay.
That is the oddball.
Okay.
You guys can read the chapter better. Anyone ever hear of a carnivorous sponge? There are deep sea, and I don't have any pictures here. I know in past lectures I did feature them, but because they're featured in the chapter, I don't have a slide on them today. But the carnivorous sponges are way cool in that they are actually predatory. They have no choanocytes. They have evolved these predatory habitats where they're feeding on small crustaceans in the deep sea. Really, really interesting feeding mode that evolved. And then the loss of the choanocytes, typical feeding cells. And if nobody was going to give that one, that's one I was going to give, because every time I read about it, it's just so cool the way you have a really unique feeding ecology that evolved within the sponges that is very different than what all the other sponges exhibit. Anything else? Any cool features about placozoans? Placozoans, what? There's a paragraph to it at the beginning of the chapter. This one discovered. Not much on that. Nothing? Okay. Remember, on Monday we're going to cover cnidarians. So read through the chapter so you have a few interesting features. It's usually the last thing I think of before the lecture. What is the interesting feature that I'm going to present, or interaction that I have with these things? All right. So nobody's had a unique interaction with the sponge? No. Anyone see a sponge in the wild? Yeah. Got one freshwater or marine? What you saw? Okay. Be ready next time for some experience that you've had with the focal group that we're going to be discussing. Because this slide, not exactly the same pictures and titles, but similar slide, similar question, every lecture. All right, so what are sponges? It's the phylum Porifera. And I have shot. Phylum Porifera. Should note in chapter 8, Porifera is a pretty easy one. Por, literally a pore, an opening. And fera is Greek for to bear. And so Porifera means pore bearing. And these are animals that are covered with numerous pores. The book says that the group contains about 5,000 species, but if you look at more up-to-date numbers, there are somewhere around 9,000 species. The vast majority of these are marine. There's just one family of freshwater species with it. Around 180 species within it. They exhibit just the cellular level of organization. With multiple cell types, so different cell types that are performing different functions. What this means too is that they have no true tissues. Last week we talked about early development and formation of early embryonic tissues, the endoderm and the ectoderm, endo interior, ecto exterior. Those are not present within sponges. When do those tissue types first originate? What stage of development do we see the development of these, the origination of these embryonic tissues? Yeah.
When it gut develops?
I can't hear it all. It's too far away. I'm
When
sorry.
it gut develops?
When the gut develops, right, exactly, during gastrulation. That's when we get two different cell types that form. Most sponges are not got to go through a gastrulation stage. They do not develop those early embryonic tissue types. In terms of other features, they have cell types, these choanocytes. Choano meaning collar, site meaning cell, shown here, that would resemble unicellular, multicellular, or sister group, the choanoflagellates. So the cell, choanoflagellate, these are choanocytes of a sponge. Cells, some cells, at least not all, but can exhibit totipotency, meaning they're kind of all powerful, kind of like embryonic stem cells. They can develop into different cell types. Question?
Yeah, so I was wondering, so like there's 9,000 species, but then how come like you highlighted the ones?
Because most of these are just, most of them are in, and many of the phyla we'll discuss.
Oh, that's freshwater.
Yes,
Okay.
I will use all kind of crazy abbreviations. Freshwater, FW. SPP is an abbreviation for more than one species. If I just did SP, that equals one species. You spell it out. I'll use lots of abbreviations, and some are standardized to me, and may not be standardized to you all. Feel free to ask as I'm going through this. So the cell types exhibit totipotency. One interesting feature, that again, let's see, I know they mentioned this in the chapter. You could take a sponge and push it through a sieve so that you're separating out individual cells and then leave it come together to form a new individual. So the cells can reorganize. Something about cells in organisms that have tissues, too, true tissues, is that there are junctions between cells. That cells are adhered to each other through these cellular junctions. Sponges don't have those things. So they can come together, but they don't have any fixed attachment between cells like we see in other animals that have tissues, true tissues. And then sponges, as an animal, have a very unique feeding mode. They have no gut, they have no mouth, they have no anus. They filter water through openings, and then nutrients are phagocytized by cells and so brought in to the animal via that mode. But they have no true gut. And the presence of all these choanocytes moves water through the organism at quite some rate. We're going to attempt to do this with some of our sponges in the lab, but oftentimes the sponges have to be happy to do what this little video will demonstrate. And here is a diver by a large hemosponge putting some dye around the base of it through the choanocytes that are active in the interior. The sponge is covered by pores. It's drawing the water in through the tissues and ultimately will emerge out this central region at quite some rate. That's all due to the action of these choanocytes. Amazing. Yes.
So, is the total potency or last or whole, is it only when it turns normal, or the cells always
Typically, we'll discuss these in a bit, but there are, one of the cell types are known as amoebocytes or archaeocytes, and those cells typically can be modified into other cell types. The choanocytes can actually be modified into sperm and in eggs in some cases. So there are a variety of different cell types can turn into different things. So there is no limit on it. The archaeocytes are always present, and those can give rise to other cell types. But not all cell types in sponges have that power. All right, so this is the tree that our text uses. And go way to the top, this is where Porifera is. And if you notice, this is kind of the common ancestor of all animals. Porifera shares that common ancestor with all animals, but all the rest of the animals share a common ancestor that is unique to them. So sponges, and this, for this phylogenetic tree, we would say that sponges diverged earliest within the animals. That's what this tree is showing. And all of these other species share a common ancestor here. In phylogenetic speak, we may say that the sponge phylum is the most basal. It comes off at the base of the tree. Any question? Yeah. So, for a long time, it was assumed that sponges did not undergo any gastrulation. We do not see any formation of true tissues in adult individuals, and it was long assumed that no sponge underwent gastrulation. But there are a few reports of a few species where it does appear that the embryonic stage goes through a gastral stage. So there are a few, but it's not widely known in sponges, and it's only recently known in a few species. Good question. So, I guess based on what we talked about today, and what we've talked about in previous lectures, why is this tree reasonable to put sponges as the most basal group? What evidence could you use to support that? Yeah.
Multicellularity, but not in the fact of having tissues and organs.
Very good. So, and I think that's what I say here. Or another way to consider it is, why might all the other animal groups be descended from a common ancestor that is not shared by sponges? Is because that common ancestor evolved tissues. So all of those species share a common ancestor, or all of those phyla share a common ancestor unique to them, not shared by the sponges, because there are new features that evolved in that common ancestor. So no true tissues. Any other features? some sponges that exhibit gastrulation, like we just talked about, but that's another feature that all other animals exhibit. Gastrulation that is unique to their common ancestor. Also, a third one is that all those other phyla that share a unique common ancestor, if this view is correct, lost the choanocyte cell. They don't have collar cells like we see in sponges. I hope I did, is not saying, well, sponges are more similar to choanoflagellates, so they're the most basal group. I didn't want to say that, because those are two ancestral characters that I'm using to say sponges are most basal. Instead, the arguments I made are to say what makes this group have a shared common ancestor, the fact that many features evolved in it. In 2008, and we have a colleague from the Department of Ecology and Evolutionary Biology is a co-author on this paper, a big phylogenetic study of animal relationships came out, and kind of shocked, at least those who care about animal relationships, but shocked the community based on this hypothesis. They find that, not sponges, but ctenophores, and I realize we haven't gotten to ctenophores yet, but that ctenophores are the most basal group. Ctenophores undergo gastrulation. Ctenophores have true tissues, they even have nerves and muscles. Sponges don't have, that all other animals have. And the support from that is, this is the tree that they had from back then, and I zoom in on this bottom part where we have the most basal members of that group. These are the outgroups. These represent non-animal eukaryotes. Saccharomyces cerevisiae is yeast, brewing yeast. And you can see on here, this is the common ancestor of all the other animals that include sponges. And it's the ctenophores that are most basal. Why did it shock us? Because what this suggests is that tissues gastrulation evolved here, and in all the non-ancestors, or all the non-relatives of the sponges. Or it was evolved here, and then sponges lost it. Complex features can be lost in evolution, but this requires multiple steps to explain the origin of true tissues, gastrulation. So it was quite a shock. A few years later, another group of authors took data sets, and depending on which data they used in their tree reconstructions, they came up with different phylogenetic hypotheses. This first one puts placozoans and sponges together, and all the rest of the animals in its way together. That's one. The other, though, supported what the previous tree showed, in that ctenophores were the most basal group. If they combined all those data, sponges were, but if you notice here, sponges were paraphyletic in this particular tree. There have been a few other studies since then. Most recent ones seem to very strongly support sponges being most basal, but it's still controversial. There are still people doing work on this question. We'll come back to this when we talk about ctenophores next week. But here's a question for you. If these molecular phylogenetic data are kind of inconclusive with regard to who's the most basal animal group. What other data can shed light on this question? Yes?
Well, we could use morphological data, but, like, I think the molecular data is more accurate.
Do you? Molecular. So he suggested you could use molecular data, or morphological data, but said that molecular data were more accurate. And the morphological or the developmental data, I think, are previous interpretations about phylogenetic relationships. So those do support the basal position because we have gastrulation, we have those different features that are shared amongst animals that aren't shared by sponges. And so that suggests they got a common ancestor. So phylogenectomies could work.
Just like as like a follow-up, like the morphological data, you could say sponges haven't developed these things, and as a result they're basal. But I think molecular data is better because sometimes we don't read morphological data the same. We view two species of butterfly as the same because they look similar, or two species of arthropod the same because they look similar. But with molecular data, our perceptions aren't a factor. It's just whether they have the same, like, DNA.
Yeah, those are good points. So he's arguing that molecular data are kind of unbiased, but morphological data could be biased by the observer. That's exactly the case because with molecular data, we have an A, G, C, or T. We're not going to be biased in how we call that character state, but in morphological data we could. So a molecular tree could be better than a morphological tree. Any other information we could use to evaluate this question about who's the most basal animal group? Yeah.
Fossil record?
Fossil record. One way. And 2015, so sponges have a fossil record. We talked about this at the very beginning class. Someone introduces. They have a fossil record that goes back to the Cambrian, also possibly in the Precambrian. In 2015, fossils were found in the Precambrian, so about 600 million years ago. So these place an earlier date on the first recognized fossils. Just this past, it's either summer or fall, I forget when I saw it. There was a paper that also came out that found these little microstructures in 890 million year old rocks that resemble what spongin fibers look like in modern sponges. One of the interpretations, and you can see they're kind of loose about it, possible sponge body fossils in early 890 million year old fossils. So the fossil record of sponges goes back maybe 600, possibly as early as almost 900 million years ago. So they have a very deep fossil record. They may have been one of the first groups. We don't see fossil records of ctenophores predating the Cambrian. Anything else? Time machines, nothing sci-fi, but anything else that could be used to investigate this? I'll show you a short clip that describes a way. And if you remember, when we were looking at early evolution of different animals group, showed a tree that showed biomarkers present very early for sponges. This is where those biomarkers come from. I'll tell you about it.
There is a specific moment in Earth's history in which most animals took over the planet, known as the Cambrian explosion, which took place approximately 540 million years ago. While sea sponges have been widely thought to be the first animal on Earth, recent genetic evidence suggests that the more complex comb jelly might claim this title. But new research by MIT scientists may side with the sea sponge. They looked at a molecule found in rocks that are 640 million years old and confirmed that it was originally produced by sea sponges, 100 million years before the Cambrian period began. The evidence suggests that sea sponges may indeed have been the first animals to inhabit Earth. Very few fossils exist from well before the Cambrian explosion, making it extremely difficult to determine what the first animals were like. So researchers at MIT have been looking for the answers in molecular fossils. which are trace amounts of molecules that have survived in ancient rocks long after the actual animal has decayed away. The researchers focus on 24-isopropylcholesterol, or 24-IPC for short, a lipid molecule that is a modified version of cholesterol. Because this molecule has been found in rocks that are 640 million years old and is produced today by sea sponges, it could be the key to unlocking evidence of the oldest animal life. In this study, the scientists analyzed genes and lipids across large groups of organisms, including multiple types of plants, fungi, and sponges, trying to determine how many times the ability to produce 24-IPC has evolved. They identified a gene responsible for this molecule and discovered that the number of gene copies each species has can predict what kind of molecules they can produce. They found that only 2 organisms, sea sponges and algae, have enough copies of this gene to produce 24-IPC. No matter how they manipulated the timing of the evolutionary tree, the researchers found that sea sponges evolved the extra copy of the gene much earlier than algae, and they did so around 640 million years ago, the same period in which 24-IPC was found in rocks. Their results provide strong evidence that the simple sea sponge, and thus animals, appeared on Earth much earlier than fossils currently suggest.
That's from a few years ago, so the other fossils that suggest 890 million, that just came out this past year, so. But again, from that, they notice an organic molecule, a lipid, present in 640 million year old fossils. Who's responsible for producing that? Do a study of different organisms, including sponges, that could have contributed to that. Based on reconstruction of gene duplication events in sponges, apparently would have evolved early enough to account for the presence of IPC.
So like, if you went to the fossil record and you know you said sponges have like one of the earliest fossil records, wouldn't that be kind of hard to say for metaphors, because they're like really having hard part and stuff that was fossilized?
So, good question. So, and that's a case in point. Ctenophores may be absent from the fossil record because they're soft-bodied forms. Actually, there actually were ctenophores. I think beginning of the Cambrian, maybe right before the Cambrian, there were hard-bodied ctenophores. I forget the exact dates on those, but early Cambrian possibly. And they did have hard parts, or at least the interpretations are that they had hard parts. But that's a good point. Ctenophore fossils may be absent from the fossil record just because they're soft-bodied and less likely to fossilize a sponge. Yeah. So that's an argument if you were supporting the hypothesis that ctenophores evolved first. I come to you and say, well, sponges have an older fossil record, you can say, but ctenophores don't. So it's a different. That's why this is so controversial. The presence of sponge fossils during the Precambrian provides evidence that they existed early during the evolution of animals, but it doesn't preclude that ctenophores were present at that point in time too, just because you haven't seen them. The absence of evidence is not evidence against something. All right, so we'll again come back to this question once we introduce the sponges. But otherwise, some aspects of the biology of the sponge in terms of their morphology and their cell types. I mentioned earlier that there are a number of different cell types present. We've talked about these already. Here's a diagram of what a choanocyte looks like. Again, a flagellum, the collar on the cell itself with these little microvilli that are supported by cytoskeleton. Movement of the flagellum causes water to move through the collar where food particles can be filtered out. So these are the feeding cells of most sponges. Panacocytes typically occur and almost form like an outer skin layer. Sometimes they're internal within certain sponges as well. But they form almost like an epidermis, an epithelial tissue around the surface of the sponge, porocytes are cells that have an opening through them that allow movement of water from the exterior of the sponge into the interior, known as the spongiceal. The archaeocytes, amoebocytes, are typically in between the two layers formed by the melanocytes and the canapocytes. In this region referred to as the mesohyl. So within that we have archaeocytes or amoebocytes, and then the various structures that are produced by these archaeocytes. You see here we have a spicule. So in the mesohyl, spicules, other extracellular matrix materials such as collagen, modified collagen, spongin, so forth. And that's what gives the sponge its structure. Amoebocytes, archaeocytes can form sclerocytes, which are the particular cell types that are producing the spicules. Spongocytes that produce sponge fibers, and cleancytes that are producing the collagen. So we have layers of cells, glanocyte layer and canapocyte layer. Glanoderm and canapoderm. The spicules that are produced by different sponges have different structures. They also have different compositions. Some produce calcareous spicules, one class of sponges, the class Calcarea. It's an easy one. It's well named. Calcarea is the one that produces calcareous spicules. Glass sponges and demosponges produce siliceous spicules that have a variety of different shapes. The siliceous spicules of glass sponges tend to occur in, the rays occur in multiples of 6. Hexa for 6. Hexactinolita is the class of sponges that are the glass sponges that produce these siliceous spicules that have 6 rays. And then there are spongin fibers that we saw in the earlier figure that suggested those microfossils were actually fibers of sponges from 890 million years ago. Every time our text introduces a particular phylum, well, at least most phylum, it didn't do this for Placozoans, it will show a phylogeny of the major groups within that phylum. Within the sponges, we have these 4 classes. And these trees are very useful because they show features that are either unique to each class or that are shared among multiple classes, at least based on this particular tree. So there are 4 main groups. This is a phylogeny that actually supports sponges as a sister group for all other animals, so being the most basal group in 2017. I'm using it here because they have a number of sponge species within it, and those first thing that's important, all the sponges come out together and share a common ancestor. And then, as represented in the tree that we just saw, hexactinolids and demosponges are close relatives, and calcareans and homoscleromorphins are closely related. So we're getting the tree shown in our text is supported by molecular phylogenetic results. Sometimes it is, sometimes it isn't. or at least the most up-to-date molecular results. So in terms of these groups, talk about demo sponges first. Demo kind of, think of demographics. It refers to, I think it literally translates to common people. So this is given for this particular group because these are the most common sponges. I started by saying that sponges of around 9,000 species. This group contains vast majority of them. It contains about 7,800 species. I have no eraser. You might be right here. So this is most of the species. Most of these are marine species, only about 180. Spongilids, a particular family, are the only freshwater sponges. They occur within the zebra sponge. They have siliceous spicules. They have spongic fibers. These are the bath sponges that are relatively soft. So they exhibit a variety of different morphologies. The Hexactinolita, these are known as glass sponges. We're not going to have any live glass sponges. In class, we'll have sponges, but we don't have any glass sponges, but we do have some glass sponge skeletons to observe. Most of these occur in deep sea habitats, so we don't have any live specimens in class. They're the only sponges that typically exhibit a radial symmetry, although some body plans of calcareans exhibit that. An interesting feature about this group is that they have a syncytial body. We talked about syncytiality last week, syncytial together cells, so multiple nuclei surrounded by a single plasma membrane, right? And it forms a unique structure that we'll see shortly that is very different than what we find in the others. It's referred to as trabecular reticulum. There are multiple platforms formed by the syncytial body and reticulations, connections in between them, places for water to move through, and places for the collar cells to occur to cause the water movement and to filter out food particles. So it's a very unique body type that they exhibit that's not shared by any of the other sponges. Group includes around 700 species. Obviously, this is the only freshwater group, so these are all marine species. Calcareans, calcium carbonate spicules. Calcium carbonate, same thing, comprises shells, the mollusks, and so forth. Relatively small sponges. I always hope to get some calcareans in the lab, but this year we don't have demo sponges. And then the final class, Homo scleromorpha, which literally means same size spicules. They typically have very small spicules, or in some species, spicules are completely absent. Previously, I think when I learned about these were placed in demo sponges, considered to represent a distinct group. And what's unique about them, and what makes them very distinct from the other sponges, is that they almost have true epithelial tissues. A true epithelial tissue is one that has an extracellular matrix that forms part of the basement membrane of that tissue, collagen and so forth, and the cells are connected to each other through cell junctions. Homo scleromorphans have a basal lamina. They also have one of the two types of cell junctions. So they don't have true epithelial tissues, but they're almost there. They have some components of this. This is an interesting feature because this is something that may have evolved within sponges and within other animal groups. Or it may have been evolved from an ancestor of all animals and lost by the other groups. Those are the four main groups. We're largely going to see examples of the demon sponges in class, including some dried freshwater specimens that have reproductive parts on them, so, or at least reproductive stages on them, I should say. Sponges have a variety of different body types that are associated with how water flows through them. Our book introduces the three main types that I'm showing here, and there are also two additional types that are the most recent edition of the book talks about. Previous editions do not. These are the asconoid. The three main ones that have been recognized for quite some time are the asconoid, cycanoid, and leuconoid. Asconoid is rather simple. Asconoid means, in Greek, a skin bag. These are relatively small, flimsy sponges. Particular body type is only found within the Calcareans, and it's very reminiscent of that first image I showed, where I showed the pinacoderm and the choanoderm. The pinacoderm on the periphery, the choanoderm in the interior. Water comes in through pores, the porocytes, through the action of the flagella on the choanocytes, and that's where filtration of food particles takes place. The simplest of body types. Only exhibited by some Calcarean species. None of the other groups exhibit this particular body type. The cycanoid is a little bit more advanced in terms of complexity. And I kind of view it as if you took one of these and you put multiple ones along the edge. Another way to view it is if you took this and you just made infoldings into it. The infoldings allow for much greater surface area, so it's much more efficient for moving water and for filtering that water. So in this case, the choanoderm lines these canals in the interior. Water is brought in through pores and filtered by the choanocytes. The cycanoid is Greek for fig. And I assume this is based on resemblance of these to a fig. You ever cut a fig in half? Maybe this somehow resembles that, or at least it did for the people who named this particular body type. The third major class, so this one is only exhibited by Calcareans and some hexactinolidans. The other type, all classes exhibit this particular body type, leuconoid. And leuconoid, you think of leukocytes, but leukocytes for white blood cells. Leuco, in this case, refers to the disease elephantiasis, where you get these huge swellings and lymph nodes and so forth, because of the hardened structures and the thickening of this material, presumably resembled the condition that one gets when they have elephantiasis. Here, what we have are canals and chambers within where the choanocytes occur. With the asconoid type, how does the water flow? It's relatively simple. In through the porocytes, and then out through the osculum. The cycanoid, the fig-like ones, coming in through the dermal ostia. See the presence of these pores all throughout. These in-current canals are lined with anachyderm. And you can see in this case, the interior of the spongiocele is lined with anachyderm. They're brought through pores, prosopods, into the chambers where choanocytes occur, leave into the main spongiocele via these other pores, apophyles, and then emerge from the osculum. The leuconoid type merging through the ostia that you can see present on here into the incurrent canals, the flagellated chambers, and then out through excurrent canals, and then through an osculum. Leuconoid types typically have multiple osculum associated with, so multiple large openings, whereas the other two types tend to have a single osculum. New types were described 11 years ago now that are slight variants on the leuconoid type. Leuconoid 1 is a figure from this paper that we find in all 4 classes. The syllabid means Greek for collect. Based on how the water is collected, it comes in through these incurrent canals, and then the chambers are arranged around single excurrent canals, as opposed to in the leuconoid, they're more equally distributed. In this case, they tend to be concentrated around single excurrent canals. You see calcareans and hexactinoidans with this type of body type. Solenoid, solon for tube. The choanocytes essentially lining these canals within the sponge body itself. So there are a large series of tubes running through these that ultimately exit into the spongiocele. So they're just slight variants. Those we only see in calcareans. Calcareans have all the different types of body types. Hexactinoidans have a couple, 3 of them, and the others just the loop body type. In terms of reproduction of sponges, we talked about cells being sort of potent. We talked about the fact that we could separate cells out and that they'll form a new sponge. There's a lot of fragmentation that sponges can undergo to form new individuals. That's kind of represented here. A bud forms, that buds can be knocked off, develop a new sponge. Or people who, I think I read this in the text, people who rear fat sponges, they sample an individual and break it off into 2 pieces and attach it to something and then put it back into the ocean and let those individual pieces grow. So they're very easy to fragment and develop new individuals from. Freshwater species and a few marine species are capable of producing overwintering stages that are asexually reproduced. The interior contains amoebocytes, the exterior spicules for protection. These can overwinter, at least in the freshwater forms. I assume in marine forms they're just meant as a resting type A that can last for some time. But the freshwater forms tend to die in the fall. They produce gemmules that can last through the winter and then develop into a new sponge. The archaeocytes in the interior emerge and form a new sponge. Question?
Yeah, I feel like this is like a bad question, but like what determines whether a sponge is alive? Because obviously the sponge, maybe not used like in the kitchen, is dead, but then it can be grown into a live sponge. Like what determines whether it's alive or dead?
So the typical bath sponge is dried out and the cells will not reanimate. So if the individual cells are actually physiologically processing and they're functioning, that would be alive. But you could not take, say, a bath sponge and get it to come back to life, reanimate, right? So the cells have to be functioning. So the cells have not ceased to exist, I guess. It's a good question, interesting question. So these cells, are these gemmules, these reproductive stages, it's asexually reproduced, but the cells inside, honestly, I do not know if they're in a state of animation where they're not doing much. How much oxygen do they need and so forth. Many animals can go through states of suspended animation where they're still alive, but their metabolism and so forth is very, very low. I'm not sure how active these are. That's a good question. How active are the amoebocytes and gemmules? What are they doing while they're overwintering? But those are stages of asexual reproduction. Sponges tend to be monoecious. They're capable of producing both male and female gametes in the same individual. Sperm are typically produced via modifications of the choanocytes, the collar cells. They're released, they're spawned from the individual. Eggs may also be spawned, but many sponges brood their eggs, so they keep their eggs within the body. And then the sponges, or the sperm, are engulfed by a collar cell and delivered to eggs within an individual. That eggs are fertilized, and then embryo develops after cell division, forms, if it's hollow, this antiblastula larvae. Remember, it resembles a blastula we talked about before, the development stage of animals, with ciliated cells. They're not known for very long distance moving, but they can exist in the plankton for a few days, and then it can settle out and ultimately develop into a new sponge. There are also larvae that are solid bodies. These are known as parenchyma larvae that have a gelatinous mass in their interior. I can spell that better than I can pronounce. Literally means solid body, and they have a solid body in terms of the interior is not hollow like an antiblastula. No question?
So is the same individual producing the egg and the sperm?
Same individuals. My understanding is that they're simultaneous hermaphrodites. So the same individual is capable of producing both sperm and egg at the same time, and then they're released. Is there self-fertilization within sponges? I do not know enough about reproduction of sponges to know if there's any way that they limit self-fertilization. It could, especially in sponges where they are releasing both sperm and eggs, there's a potential for that. So there may be mechanisms that prevent self-fertilization. Within those sponges that are brooding their eggs, if they absorb, engulf their own sperm, is there a method for preventing self-reproduction? I don't know either. Presumably there are. Many organisms are means by which selfing is prevented. You can have gonads we don't have gonads, we don't have true tissues in these beasts, but the cell types, sperm and eggs, can be developing at different points in time to prevent self-fertilization as well. So those are good questions. I don't know the answer. And we will have, I know we have some gemmules available in the lab on slides to observe, and we have some freshwater sponges, some dried freshwater sponges, that they must have been collected in the fall, because you could see the gemms on the bodies of these sponges. And then we could take them off and look at them under a scope. You haven't had them in water enough to see if they would reanimate after, God only knows when these were originally collected, but they must be old. I don't think they would reanimate, but the geoscientists can do that experiment. All right, so sponges are a major source of pharmaceuticals. And a lot of people see sponges as having very few defenses, so evolving unique compounds that prevent predation or infection may be particularly important in sponges. So they've also been a major target of people interested in designing drugs. Just past year, 2020, a few different compounds identified from sponges that appear to be useful in combating cervical cancer. Some are anti-malarial, an antiplasmodial compound from this particular sponge was identified. There are compounds from this almost automorphin that affect viral loads for patients who have HIV. And illustrated here, you have a viral load within a patient, and then apply some antiretroviral treatment to them. Viral load drops. You do nothing more, it eventually goes back up again. But if you treat them with this compound from the sponge, it's cortistatin A, it tends to keep viral loads continually low. Who's heard of remdesivir? Nobody's heard of remdesivir? What? Really? Oh. Remdesivir is a drug that's used for treating COVID-19. It was derived from a compound that was extracted from a sponge in the Caribbean, the Zima sponge, back in the 1950s, that was found to interfere with viral replication. How can you interfere with viral replication? One way to do it, and this is how remdesivir acts, but how these compounds work is by interfering with replication of retroviruses, of RNA viruses. Remdesivir was developed from so it's not the original compound that they discovered in the sponge back in the 50s, but it was developed from this compound. It was slightly modified to act on particular viruses. Various drugs from this that exhibit antiretroviral activities have been useful in treating HIV patients, and I think I've listed others. COVID. And how it works is pretty cool. So remdesivir is actually given to patients as a drug, so it's not in an active state when it's first given. It then becomes activated. And if we notice these three little phosphates here, if we know anything about replication of nucleotides, whether it be DNA or RNA, it's relatively similar. We need a nucleotide triphosphate. How many triphosphates? Here. I assume you cover this in genetics as well. So a nucleotide triphosphate, an ATP, a GTP, a CTP, the other one, TTP, right? Or for RNAs, the RNA equivalents. What this particular molecule is it's a nucleoside analog. So it looks like a nucleotide base. And in this case, it has ribose associated with it. So it looks like an RNA base. In coronavirus, when you introduce remdesivir to patients, it interferes with the replication of the RNA, this transcription of the viral RNA, because this little molecule gets incorporated as a base, and then it prevents the ultimate extension of that molecule. So it prevents replication of coronavirus and other types of retroviruses. So before we move on to placozoa, I have enough time to complete that. Any questions about sponges? Four main classes, a couple different body types, and how water circulation patterns in terms of the organization of tissues within. One of the major things I think facing zoology today is the potential basal position of sponges or ctenophores. Question?
Yeah, I guess I was about sponges. Actually, the sponges have lost their color, so how would they?
Good question. I do not know. So they don't have monocytes. I would guess there may be amebocytes present within them that produce the sperm, but I do not know. It's a very good question. I'm not an expert in the biology of the carnivorous sponges, and they're very deep sea, so I'm not sure if it's how reproduction proceeds in them. Great question. Any other questions? All right. Placozoans. If anyone finds placozoans in our tanks, you'll get bonus points. Placozoans are such a cool phylum. For the longest time, up until just a few years ago, there was one species. Trichoplax adherens. Trick hair, plax plate, hairy plated adhering things. And they were discovered over a century ago in the 1880s, growing on the sides of a marine tank. Little tiny things. I assume somebody looked at what the hell are these little tiny things on? Put them on a glass slide, looked at them, and saw things like this. Very small. They're around 1 to 2 millimeter in size. Just right out of space. A few thousand cells. Very simple construction to them. They would feed by going over top food material, secreting enzymes to digest it, and then phagocytizing that material into the body. Very simple beasts. They don't, like sponges, they don't have any true tissues. They also don't exhibit any types of symmetry. When they were first looked at, and I don't know if it's because people had cnidarians in their tank, but they thought maybe these were cnidarian larvae that had settled on the side of the tank and just get metamorphosed into cnidarian. They're very simple. They have a few different cell types, which we'll see, but they're very simple. Also for all animals, I'm not sure if this includes some of the parasitic animals that produce genomes, but they have one of the smallest genomes of any animals. Relatively simplified. Look in terms of what makes them up. The hairy, the trichos, they have cilia that emerge from cells. Upper level, they have what are referred to as epithelial cells. It's not a true epithelial tissue though, because of the basal lamina and cell junctions not being present. They have gland cells that are secreting various enzymes, lipophil cells that are secreting enzymes that are associated with food digestion that occur on the dorsal or lower surface of these They have different type of cell type, the ventral or lower surface. What else am I missing? These crystal cells that they tend to have on the periphery that are apparently associated, they're sensory structures capable of sensing gravity, or at least that's what's suspected. And then in the interior, they have these fiber cells. So it's different cell types, no true tissues within them, very simple organisms. Another view of the diagram is to show the additional cell type. So there have been different gland cell types that have been identified, but also these shiny spheres. They're not really cells, but they're droplets of lipids. And because if you present a Placozoa to a snail or something like that, it tends to run away from them, as snails do. It looks like these may be little defensive structures that are present. They reproduce, sponges do, by simple fission or budding off of parts of the beast. It looks like they're capable of producing sperm and eggs. These are presumably oocytes present within an individual. Some of these workers, Shearwater has been able to produce embryos, but they never last very long, so we don't know much about its life cycle, its development. But presumably, it develops into a young Placozoa. In terms of their phylogenetic position, Archery kind of puts them here in a polytomy with cnidarians and xenovores. And most recent phylogenetic reconstructions of animals that I've seen aren't really sure about where they lie. They may be a sister of sponges, or they may be a sister of the rest of the animals. It varies depending on which data you're looking at and how you look at them. But we're not sure where they fall out. A few years back, 2013, a group of researchers looked at DNA sequences from individuals collected from various places around the world. They found a lot of different clades. At this point in time, we recognize it's one species, Trichoplax adherens. This was kind of exciting because it suggests that there's a lot more diversity present than what was anticipated. There may be more than just one species. This is related to that. So you see 7 different clades here that may represent 7 different species. This is where they occurred in different geographic regions. So you can see a lot of colors, like this magenta color, all throughout the Mediterranean region, also in the Eastern Pacific on the west coast of the U.S., in the Caribbean region, in Hawaii, in the Indian Ocean. Some are very wide distributions. You also get a number of different clade types occurring in the same location. Morphologically, they're quite boring, but there's perhaps more that's going on that we don't know about. Since then, so this is the original species we knew about. Since then, another species has been described. This was in 2018. This one here, do you see how different it looks than Trichoplax adherens, right? Genetically, it's very different. But morphologically, it's quite the same. That was a joke. What they did discover is this weird form here. Polyplacotoma mediterranean, found in the Mediterranean, that not only has a different morphology, but is super large. It's about a centimeter in size. It's about 5 times larger than the largest Placozoa previously observed. So another new species that was just recently described, 2019. We go back. Not going back. Oh, I see why. My Google Calendar buffalo. We add those things to the tree. You see these are the original clades from that earlier tree. This is the more recent tree. developed from that previous tree. This is where that Trichoplaxogenes individual that I showed is. This is that new species that was described. The Polyplectoma mediterranea, the weird one, is the most basal, according to this tree here. So all of these others may represent additional species that also need to be described. So Placozoans are a cool group. Our book doesn't really do them justice. There's a paper from this fellow Shearwater a few years back that describes his fascination with Placozoans. He spent his career studying these, and he was inspired based on some of the early things he learned about them as an undergraduate or graduate student. So, I got two sponges and Placozoans. Again, we're not going to see any Placozoans, but if anyone does find one in a tank, I doubt if you will, because the tanks have only been set up for a couple of weeks. Oftentimes they're found in tanks that have been set up for quite some time. And where you're bringing in material from the external environment, which is not artificial seawater. You can find these from the tanks. They're difficult to find in nature, but Shearwater has found a variety from different places. All right, so we have sponge lab this week, tomorrow, and Friday in the session here at